Lovely. Well, welcome everyone. Thank you for joining us on our first Facebook Live this afternoon. This is the first 2023 Cure Cancer Lunch and Learn with our very new fund of researcher we've just announced on World Cancer Day, Dr. Heather Murray. Welcome, Heather.
Thanks for having me.
Pleasure to have you here. I know that we have many of our donors, our supporters, we have some of the Cure Cancer Board, our team, committee members, and our observers here as well. So thank you so much for being here and taking part in this session. I hope you find it enjoyable and informative. If you would like to ask some questions, just drop them into the event chat, and we will be weaving those into our conversation as we move through the afternoon. The session will take approximately 45 minutes as well. So please feel free to ask questions. We'd love to have your input. Unfortunately, we can't all do physical lab tours, so this is our next best thing. But please feel that you're free to ask questions as you would like as well. So over to Heather. Dr. Heather Murray is one of our 2023 grant recipients. She's based at the University of Newcastle and Hunter Medical Research Institute. Heather is researching a type of blood cancer, which is called AML, or acute myeloid leukemia. AML is a disease that affects the blood and bone marrow and is one of the most aggressive blood cancers, sadly. AML affects an estimated 200,000 people worldwide each year and is responsible for 120,000 deaths annually. Despite advances in treatment, the long-term survival rate for AML remains low, with just under 30% of patients surviving 5 years after diagnosis, which is, quite frankly, a terrible, terrible result. So this really underscores our need and Heather's needs to continue to research this disease and the efforts that she is putting in place with her team to improve our understanding of this disease and develop new ways and treatments to improve patient outcomes. So thank you, Heather, for coming here. Obviously, you're new to our funding, and we're delighted to have you as one of our new 2023 research cohort. For those of you that haven't met you before or know anything about you, do you want to just share a bit about Heather and why you started in cancer research?
Yeah, hi everyone, thanks for joining. I'm Newcastle born and bred. Newcastle's about 2 hours north of Sydney. And I always loved maths and science subjects growing up at school. When I finished high school, I didn't know what I wanted to do as a career, but I knew I wanted it to involve maths and science. So then I looked at what degrees I could do, and I decided to undertake a Bachelor of Biomedical Science here at the University of Newcastle. And it was during that degree I got to hear about the research that was happening here at the uni, and I just became fascinated by the different ways that different problem-solving techniques could be applied to biological problems to improve health outcomes. So at the end of the second year of my degree, I got the opportunity to do a summer scholarship, which meant I was able to spend the summer working on a research project at the Hunter Medical Research Institute, which I loved and decided I wanted to pursue cancer research.
Yeah, look, it's really important. We're finding that less and less people are coming into research as well. When you started in research, was it challenging to enter the cancer research sphere, or did you find it quite easy after you'd done your PhD?
Yeah, so that is a huge challenge, finding funding after you finish your PhD. So I was really lucky that my PhD supervisor, had been writing grants to try and get some funding to keep me on post finishing my PhD. So I got a year's funding from her, and which allowed me to write some grants, publish some papers, and try and find my own funding.
Yeah, great, lovely. So we've talked a little in my introduction about AML, and sadly, it's very low survival rate. What drew you particularly to this type of blood cancer research?
Yeah, so when I finished my undergrad degree, having decided I wanted to do research, the next step is then to undertake a PhD. So I was looking at what PhD projects were available, and there was one that particularly drew my interest, and it was on acute myeloid leukemia. So drew my interest for a couple of reasons. Firstly, because AML is such a poor prognosis cancer. So there are around 1,000 Australians diagnosed with AML each year. So it's drew my interest for a couple of reasons. Cancer, but because there are around 900 fatalities from AML each year. So it's really poor prognosis. There are around 1,000 Australians diagnosed with AML each year. So it's drew my interest for a couple of new cases per year, because there are around 3,000 fatalities from AML. So even though it's really poor prognosis, the incidence of AML is a lot less common. The fatality rate is up there with things like breast cancer. So a lot to be done, even though breast cancer field as well, but the incidence of AML done in the AML field, which was one reason I was drawn in. And the second thing that drew me in was that the project was a lot to be done, even though breast cancer field as well, but to identify new therapeutic targets for AML, which was one reason I was drawn in. So that was really exciting. Second thing that drew me to me, so project was why even proteomics is a technique that allows us to identify and measure. So that was really exciting. All the different to me, president and step even proteomics is a technique that, and this is really important because to identify proteins are the major building blocks for cells and in the body. Proteins keep an elegant structure. They allow, and this is really important with each other and work. And the majority of drugs target proteins, so it made sense to me that we really need to understand what's happening with the proteins in the cancer cells, so then we can design therapies that specifically target the cancer cells and not the normal healthy cells.
Okay, thank you, and thanks for explaining about proteomics as well. For those that don't know what AML is, just in lay terms, can you describe what this blood cancer is and why it's different to other blood cancers as well?
Yeah, so there's a couple of different, there's two main ways, I suppose, to subtype blood cancers. One is by whether they're acute or chronic. So acute means they develop really rapidly. So within days to weeks, they will progress, whereas chronic, it's more months to years. And the second way of classifying leukemias is by the cell type they originate in. So that is either myeloid or lymphoid. So there's a number of different cell types that make up your blood, because your blood does a number of different functions. So you may know that it carries oxygen around your body to your tissues, carries away waste products. It contains immune cells that fight infections. So acute myeloid leukemia is a very rapidly progressing form of leukemia, and it affects the myeloid cells, which are the cells that should be producing things like platelets to help form clots when you're wounded. It produces immune cells to help fight infections, and also red blood cells to carry oxygen around your body.
Yeah, so it's quite important, absolutely. And when you say acute and it moves quickly, what? And it's always difficult because everyone's different, but what time frame are we talking here from someone having a diagnosis? How quick does AML regenerate and cause issues to patients?
Yeah, so if it's left untreated, typically it's fatal within a couple of months. It's very rapid.
Wow. And does AML typically affect a subgroup of people? Is it age related? Is it ethnicity related? Or is it quite a broad range of a blood cancer?
Yeah, so it can affect all ages, but it is skewed towards the older age group. So over 60 years is the most common age group, but it can be diagnosed in children as well. Slightly more common in males than females, so we don't actually know why for that one.
Oh, that's interesting. Okay. All right. Thank you. So can affect everyone, but more skewed towards people senior in years as well. And why do you think that is? Is that just because of the cells deteriorating over time? What's the thinking behind that?
Yeah, so there's sort of one hypothesis that it's because your cell can accumulate mistakes, I suppose, as you age. So as you get older, it's more likely that your cells will have accumulated mistakes that could start a leukemia.
Yeah. Okay. Thank you. So you obviously started this research and you talked about how you were interested in proteomics. How new is proteomics and the study, and where do you think the proteomics work will take you in regards to AML?
Yeah, so it's been around, I suppose, for a couple of decades, but it's really in the last sort of 5 to 10 years that the technology is really being improved to make it amenable to analysing patient samples, and it's really robust and repeatable, the results that we're getting. So I started my PhD in 2015, and we didn't have anyone doing any sort of large-scale proteomics experiments here, but during my first year, we were successful in a grant which enabled us to buy the top of the line at the time, mass spectrometer, which is the instrument that we use to run our proteomics techniques.
So super. So you talk about it wasn't really looked at. So you started that and you've been doing it for a number of years. Let's just go back to getting funding and the start of your career. We talk about it because it's very much our USP and what we do, but funding early career cancer researchers is vital. So we continue with that pipeline of researchers. How important for you was this funding, and also for your project on AML?
Yeah, it's super important. So last year when I got the news that I was successful, at the time, I only had funding to fund my research assistant part-time this year. So the funding from Cure Cancer means I can fund her full-time, have her helping me out full-time, and also buy the consumables that I need to conduct the research that I want to conduct this year.
Yeah, no, lovely, great. So can you just talk a bit about your work and your team? So how's the team set up at University of Newcastle at HMRI, and how do you, on a daily basis, what do you do when you go into that?
Yeah, okay. So I've got my own research assistant, which is awesome for someone, for an early career researcher. I also co-supervise a couple of PhD students, and I work very closely with a couple of other AML researchers here at the uni. So yeah, when I started my PhD, it was just the one group of us working on AML, basically 3 of us, and now there's 3 different research groups, and we all sort of work together and help each other out. So work very closely with the people in my team, but also the other AML researchers here, and obviously my mentor and supervisor, Nikki Barrels. And so on a daily basis, for example today, so if a patient gets diagnosed with AML at our local hospital, they can consent for us to receive a sample of their blood and their bone marrow, because they'll be having those samples taken as part of their diagnostic screen anyway, and they can consent for us to receive a little bit of the sample, and we can bring it back to the lab, as we have done for our sample today, extract the leukemia cells, and then we perform a bunch of tests on them. So we screen their sensitivity to a panel of about 100 drugs, and we also take that sample and perform proteomics on it, so we can look at what proteins are present in that sample and how much of them, and how that relates to which drugs they're sensitive to.
Great. So I mean, you've got a living, breathing example today. So I didn't actually realise it's quite so much immediate, straight from the hospital to your lab, which is brilliant. So just in a case like this, then, and they're being diagnosed with AML, then after you've looked at the blood and looked at the proteins, what's next for the patient? What treatment plan are they currently on? And I know there's differences for each patient, but what's a typical treatment plan for a patient now versus what you're trying to achieve in the future?
Yeah, so right at the moment, everything's de-identified, so I'm not allowed to know exactly what treatment that patient's being put on, and they're not allowed to know any of the results from our tests at the moment, unfortunately. So they'll undergo some screening at the hospital, probably for looking at which genetic mutations they have, and that provides a few different options for currently available treatments. So the standard of care at the moment is still really chemotherapy, which is highly toxic, and so toxic that some patients, for example, older patients, can't actually receive it. And there are a couple of targeted therapies available, like venetoclax, which is what my project centers around this year. But what I'm hoping to move to with our work is that one day, or hopefully not too far in the future, that when we take these samples from the hospital and come analyze them in our lab, that we'll be able to give our results, the information from our results, back to the clinician and tell the clinician, well, these are the drugs that this patient was sensitive to in our screen, and these are the proteins that they express, which means that they will or won't respond to a particular panel of drugs. And so we really want to personalize treatment to each patient, because really every leukemia is different, just like each person is different. So we think the treatment plan for everybody should be personalized to them.
Yeah, wonderful. I mean, imagine a world where you have a unique personalized plan according
Yeah.
to your allergies and your proteins in your blood. That would be incredible. So we've talked about a few challenges. Sorry, I'd just like to say, just remind everyone that if you do have questions, pop them into the chat there on Facebook, and I will weave them into our conversation as well. What does the funding that we're doing at Cure Cancer, and thank you to our donors here that are supporting you today, but what does that mean to you personally in your aim for your career?
Yeah, so it's going to allow me to conduct this work that I wanted to do this year, and also employ my research assistant to help me do that this year. And so I really hope, well, this funding gives me an excellent footing to conduct this work, which will put me in great standing to apply for even larger grants next year, so that I can keep funding my research and my research team.
Yeah, look, great, thank you. And obviously it's a catch-22, but you know, when you receive more funding, where do you see that going? What do you want that to be spent on, and what's your ongoing focus?
Yeah, so as I mentioned, I'd love to see precision medicine be the standard of treatment for every patient. So to get there from where we are now, we need to undertake a clinical trial so that we can prove our personalized approach is better than the current standard. And so to get there, I need supporting data like I'm going to collect this year with this grant. And I will also, I'm going to need to expand my team a little bit to help support all of the work that goes into setting up a clinical trial, and having also equipment so that we have dedicated equipment available the minute a sample comes into our lab, that we're able to analyze it straight away.
Right, and what type of equipment, just for everyone's interest, what type of equipment do you use at the moment?
Yeah, so we have a dedicated facility for cell culture and handling samples. So when we get a sample from a patient, it needs to be handled in sterile conditions. We can't just handle it on the bench, for our own safety, because we don't know if that patient could perhaps have COVID or something like that we could infect ourselves with unintentionally, but also to protect that sample from anything that might be in the air, like bacteria. We want to make sure that it's pure. So we have a dedicated cell culture facility where there are biohazard hoods. So we work with the samples in the hood, which has its own airflow, own sterile airflow. So that's where we conduct all of our assays on the samples. So we've got a robot in one of our hoods as well, which allows us to aliquot very tiny volumes of drug into plates, so that we're using as little as the sample as possible for all of our assays. So we can do maximize the number of experiments we can perform with typically sort of 5 mils of blood that we receive. And then the proteomics technique itself is unfortunately very expensive. It requires very specialized pieces of equipment, in particular the mass spectrometer. So the mass spectrometer itself costs upwards of $1 million and requires a dedicated specialized technician just to keep that machine running day to day. So salaries for people to run these dedicated pieces of equipment as well.
Yeah, no, that's a lot. And do you have one of those dedicated people at HMRI as well, or do they come? Are they further afield and you rely on their research and then bring it back to your lab?
Yeah, so we have a mass spectrometry facility here at the uni. So there are a couple of mass spectrometers and a person that runs them. Mm-hmm. But to move our work forward into a clinical trial, we'll probably require another one of our own, because the ones we have are quite heavily used by the other research groups here as well.
Yeah, understandable. So just talking about that potential clinical trial as well, you talked about what you wanted to achieve. What's the time frame on that pending funding to do so?
Yeah, well, I estimate that in about 3 years, we should be able to collect the preclinical data that we'd need to support beginning a clinical trial like that. And AML researchers here, we work very closely with clinicians, in particular Anup and Jetty, who's part of the Australian leukaemia and lymphoma clinical trials group, so the largest clinical trials group in Australia and New Zealand. So we have the expertise through our collaboration with him to start a clinical trial. After we've collected the required preclinical data, it'll just be obtaining funding that will be the roadblock moving forward.
Okay, no, it's and that's what we live and breathe.
Yeah.
I completely understand that challenge, but how exciting would it be if we could help get you to clinical trial on this as well? So from your perspective, what are your hopes for the future with your work? You've talked about a clinical trial. Are you doing any international collaborations at the moment, or is that maybe something for future?
We have a few collaborations. So during my PhD, actually, I was able to go to Belgium and collaborate with a group over there to learn some new techniques that we didn't know how to do here in Newcastle. So I was able to go and learn and bring the techniques back here. And since then, I've also been to Sydney to learn new mass spec proteomics techniques, and again bring them back here to Newcastle so that we can perform those techniques here. And yeah, we've established collaborations with a number of clinicians in Australia as well, in Queensland and Adelaide.
Yeah, look, it's really exciting what you're doing, and it's great to be able to fund you. I've just got a question here. I don't know if you can answer it, but someone has asked, how do you limit or reduce your likelihood as you age of getting AML?
Yeah, that's a difficult one. So there are some lifestyle factors that have been linked to an increased risk of developing AML. So things like smoking, exposure to herbicides, pesticides, fertilizers, industrial chemicals maybe that you might be exposed to as part of your job. Obesity has also been linked. So there's things like that, I suppose, that you can try and limit in your lifestyle that will give you the best opportunity to not develop AML.
Yeah, thank you. I mean, it's something we hear with other cancers as well, and we talk about sugar, obesity, smoking, and chemicals. So it sounds like it's very similar to some of the other cancers we have talked about too.
Yeah.
And we have here, we've got so many people watching. Is there a message that you would like to give to our audience about how their funding or their support of you is helping you? You know, I know you're very incredibly grateful, and so is your team. But, you know, is there a message, some kind of lasting words before I go to the final round of questions that you might want to tell people and about the work you're doing and the impact their donations is having on the future of cancer research?
Yeah, I'd like to say thank you to everyone that's donated and supporting cancer research. It means a lot to me personally because the work that I do is very important to me, and so being funded means that I get to continue doing that. And I mean, there was an economics report a couple of years back that looked into the benefits of medical research, funding that goes into medical research, and they found that on average, for every $1 that goes into medical research, more than $2 is put sort of back, is given back in terms of health benefits and prevention of health treatment. Yeah, so things like that. So it's really an investment for the future for everybody.
Yeah, no, absolutely. I think that's really important as well. You know, we look at the long-term implications of treatment and hospitalization of patients. And if we can avoid that, then that's also, if we're living longer with a better quality of life, but it's also avoiding those costs associated with that as well, isn't it? Yeah, lovely. Well, I wanted just to thank you very much for your time today. It's been very very interesting, and it's lovely to learn about that work that you're doing. So as I said, that headed at the University of Newcastle and HMRI, and we look forward to having you. I know you're doing a couple of events for us coming up as well.
Yeah, looking forward to it.
I know the donors are very much looking forward to meeting you in person as well. But in the meantime, thank you for taking the time out of your day to be here with us. Thank you for our donors and supporters as well. It's great to have you here. Heather is the face of our BarbeCURE campaign, so if you do see her around, the money that we are raising through BarbeCURE this year is going to fund more researchers like Heather as well. So thank you for your support, and it's great to have you as one of our cohorts for 2023 as well.
Thanks very much for having me. Yeah, and thanks for everyone for your interest and your questions.
Yeah, absolutely. And if anyone on the line has other questions, please drop, most of you have my email address, drop me an email and we'll be able to follow up with Heather afterwards. Thank you to our guests. It's been great to have you here for our first Lunch with Orlando 2023. And we'll be back next month with a new researcher talking about their area of research. But in the meantime, have a good afternoon and we'll talk to you later. Thank you.